get it to fit into your book, or turn your book sideways. This is the primary sheet that you're going to be working on. So once you actually have the other pages secured, this is what the work is going to be going on. So I recommend this being third in the lineup. Okay, so I want you to notice that on this activity, it's called protein folding. So we're going to be dealing with, obviously, folding of proteins. So when we're thinking about proteins being folded, we're talking about how are we making that final three-dimensional shape of a protein. And remember that structure determines function. So the three-dimensional shape of a protein is going to be really important for that protein to be able to function the way it's supposed to function. So I want you to notice that first off, on this top sheet, there are 4 pre-activity questions. You need to make sure to answer all 4 of those questions in your lab book. You also need to notice where it says amino acid color chart. This is not the same thing as your genetic code. So your genetic code, okay, here's an example of a genetic code. Your genetic code you can find in your textbook. Now, can anyone tell me what page number that can be found on? It's not in chapter 3.
Chapter 12.
Yes, it's chapter 12.
238.
238, what edition textbook is that? And it's 242, so it's either going to be on 238 or 242. That's for the genetic code. So this amino acid color chart is actually trying to show you something different. It's trying to show you some qualities of amino acids that we can determine from, if you remember the basic shape of an amino acid, where we've got that amino group, so we've got the N portion, we've got our central carbon, and then we have that carboxylic acid on the end. When we're looking at that NCC kind of arrangement for an amino acid, that central carbon has a variable R group on it. And that means that that central carbon can have anything at all on that central carbon. Anything. Hydrogen, oxygen, big giant phenyl rings. It can literally have anything on it, and it would still be considered to be an amino acid. So what happens to the chemical nature of an amino acid if you change the chemical nature of that variable group? What do you think will happen? What happens if we put a big giant hydrophobic group right here?
It'll make it hydrophobic.
It's going to make it hydrophobic. What about if we have a big giant hydrophilic polar portion right here? It's going to be polar and hydrophilic. What if it's an acid? It's going to be acidic. If it's a base, it's going to be basic. And if it contains sulfur, like cysteine does, then it's going to have the ability to form sulfide bonds. Oh my gosh. Guess what a sulfide bond is? What kind of bond? What kind of bond do you think it would be? It's a covalent bond. Yet again, we have to just name every covalent bond, right? So, another one of those? So yeah, so you've got a covalent bond that can form between cysteines because they both are containing sulfur on the end of that chain. So, okay, remember what I said about colors? It's really difficult to hear. So on this chart, it's called a color chart because it's trying to assign colors to the different amino acids based on the chemical nature of that side group of the amino acid. How many amino acids do we find in life forms here on planet Earth?
20.
We find 20 different amino acids. 20, 30. Yeah. It's just 20. Yeah, it's just 20. So the 20 amino acids that we do find in life forms, then you've got a genetic code chart that you can use the mRNA codon to determine what amino acid would be located there. And then this chart right here is going to tell you about the chemical nature of that side group. So if you'll notice, Let's look at the very top first one, alanine. Alanine, where the symbol is ALA. What is the property that is listed right there for alanine? Gabe?
It's hydrophobic and non-polar.
Very good. So it's hydrophobic and non-polar. What about asparagine? Asparagine. What is the chemical nature of asparagine? Neurogena.
Hydrophobic.
So hydrophilic and polar. Now those are pretty big differences, right? If you're hydrophobic or hydrophilic, then that's going to determine partly where you associate in a cell. For instance, in a membrane, do you think you're going to have hydrophobic? If you had a protein that's going to embed in a membrane, do you think that it's going to have hydrophobic or hydrophilic amino acids? What's going to allow, oh my goodness, don't forget, final exam, 2 weeks, right? What is a membrane made of? Phospholipids. What kind of chemical nature is a lipid? Hydrophobic. It is hydrophobic. So I'm going to ask the question again. What kind of amino acids do you think you would have in a protein that is embedded in a membrane? Hydrophobic. Hydrophobic. So we can see that there's hydrophobic, non-polar, hydrophilic, polar. Take a look at cysteine and tell me, Jalen, what does it say with cysteine?
Sulfide, covalent bond.
Yeah, so it's got these ability to form sulfide. That's just between 2 sulfurs. So one cysteine has 1 sulfur, another cysteine has another sulfur, and they can form those sulfurs in that side chain right there, can form a covalent bond with each other called a sulfide bond. It is a covalent bond. And then how about, let's see, glutamic acid. Carter. Glutamic acid, by the way, is also known as glutamate, not glutamine. So glutamic acid is also known as glutamate. All right, Carter, can you tell me chemical nature of glutamic acid? Yeah, it's in the name, right? Yeah, it's an acid. It's an acid, okay. And then how about, what do we see? Oh, some of these are really hard to read. Very difficult to see. Oh, up there with arginine. Daniel would. Arginine.
Basic.
Do you have copies, extra copies of this?
I do have extra copies. I've got extra copies. Some over there, and then I've got these right here. And I'm sorry, Daniel, what did you say?
Basic.
Yeah, it's going to be basic. So let's think about that for just a second. Acids and bases, you can have those coming together to form bonds. You've got disulfide bridges that can form covalent bonds between different amino acids. You've got, what kind of bonds do you think you would form if you are hydrophilic and polar? Hydrogen, not ionic, no. Hydrogen bonding, back again. And so if you notice, there are colors that are associated. So the reason it's called a color chart is because we've basically made colors that are associated with the different chemical natures of the amino acids. So hydrophobic, yellow, like butter. Hydrophilic would be white. I don't know why, because like white frothy waves. Acids are going to be red, I believe, and bases, I believe, are going to be blue. Yep. And then cysteine is going to be green, because what kind of bond does a cysteine form with another cysteine? Sulfide, a sulfide bond, which is what type of bond? It's a covalent bond. Very nice. So go ahead and make sure that you're answering those pre-activity questions. And then I'm going to tell you what you need to be working on this last sheet. And then I'm going to give you some time to actually work on this, okay? So on this last sheet where it says from DNA to protein, record sheet, for step A, that is your DNA molecule. So you've got your DNA, so that's your gene. your DNA sequence. You are going to, in step B, transcribe that gene into mRNA. So go ahead and transcribe the gene into mRNA. For step C, you're going to translate the mRNA into a sequence of amino acids. Now you're going to have to use this color chart in order to find out what color each of those amino acids are going to ultimately be, because you are going to model a protein structure. You're going to model it with a pipe cleaner and pony beads. Woo! So you need to make sure that you are first transcribing the DNA into mRNA, and then translating the mRNA into an amino acid sequence, and then you need to make sure that you are writing down the color of the chemical nature of each of those amino acids, so you will know what color of pony bead to actually add in a sequence on the pipe cleaner that you're also going to be provided with. Once you actually finish getting all of the colors figured out, so you've translated your entire DNA sequence from the transcribed sequence of mRNA, once you have that done, then you can go ahead and order your amino acids, the pony beads, onto your pipe cleaner. And once you have it just in a straight line like that, spread out those pony beads on the pipe cleaner, and that's going to signify that you've actually finished writing the answers to the pre-activity questions and transcription and translation on that last sheet, okay? Any questions for me? All right, we're trying to get the study guide uploaded by today or tomorrow at the latest. So please be on the lookout for that. That's going to show up on Infinite Campus for you, and it is going to be something that you are going to have to download and print out so that you can answer all of those questions for a grade to turn in. Let's see, how's the background research paper going?
I don't know my project. You never said anything about my project.
So what have you been doing as far as background research?
I was researching like 3 projects.
Oh no, your
I don't know
paper
if I
is
can do
due a
mine.
week from Friday.
I know.
Okay, you should have at least emailed me or something.
Okay, I was gonna come yesterday.
Yeah, it's called email. It's irrespective of subs.
Yeah.
Anyway, yeah. So you definitely need to get that locked down, because now you're down to, 8 days or something, and you've not used any class time at all to work on your research paper. That's not good.
No, I did. I have like 3 projects that like work.
Yeah, the background research is the part I'm talking about though.
I've researched like all 3 of them.
Well, isn't that good? All right, well, cool. Then let's talk about which one you should actually write the paper on.
Okay.
All right. Any questions about anything? All right, I'm going to give you about, 10, 15 minutes to try and get this done. So get it done as quickly as possible, because I would like to talk about it before we break for lunch. Okay. Can we pause the video? Okay, so let's see, I think filming has begun again. So let's go ahead and get started with the activity that you guys were working on before lunch. So you were supposed to take all of the beads that you had in your little Ziploc baggie, and you were supposed to distribute them on your pipe cleaner. So, and distribute them does mean like spread them out. So spread them out on your pipe cleaner, nice and evenly spaced. Then hold it up, I want to see it. Okay, hold up those pipe cleaners. Oh, you've already made a secondary structure. All right, so I'm happy to see these. You can't keep them in their primary structure right now because we're going to kind of step through this process. I want to do this by actually also answering the first question in the pre-activity questions, which was, what are the levels of organization in protein structure? So let's go ahead and pick up with the levels of protein structure. Rashawn, can you please tell me the first level of protein structure?
Primary structure.
Primary structure, and also give it, it says briefly describe each, so don't just list, but also briefly describe.
It's just a sequence of amino acids.
Very good. So it's a sequence of amino acids. Now what kind of bonds are holding these amino acids together? Covalent peptide bonds. Very nice. So we've got covalent bonds. And those covalent bonds, they are formed through what chemical reaction?
Dehydration.
Very good. So they're formed by dehydration synthesis. In what organelle does this take place? Inside ribosomes. Ribosomes are made of what kind of macromolecule? Ribosomal RNA. Very nice. There are proteins associated, but it's the ribosomal RNA that's responsible for the actual catalysis of the peptide bond formation. Okay, and so let's see. Ribosomal RNA is transcribed where?
In the nucleus.
Oh, good job. It's in the nucleolus. That is what a nucleolus is. A nucleolus is a region of the nucleus where the ribosomal RNA genes are located. And they are just transcribed and transcribed and transcribed and transcribed. Are they translated?
No, not yet.
They are not ever going to be translated because the ribosomal RNA itself is what becomes the ribosome. So when we talk about the nucleolus being the site where ribosomes are made, really we can specify it's the site where the DNA, the genes for ribosomal RNA is actually located and where the ribosomal RNA is being transcribed. Good job with that. Okay, so let's go back and look at then your pipe cleaner with your beads on it. Then all elongated like that. That is an example of your primary structure. So that primary structure is just that elongated pipe cleaner with your beads on it. Let's talk about the next level of protein structure. Raina, what's the next level of protein structure? Secondary structure. Briefly describe that for me.
It's when the hydrogen atoms different.
Okay, so it's when we have hydrogen bonds that are causing some specific shapes to occur in your string of amino acids. Now this goes back to understanding an amino acid's structure. So understanding that you've got an amino group over here, you've got your carboxylic acid over here. Now anytime, and remember we have final exams in 2 weeks, right? So anytime you have an electronegative atom that is covalently bonded to a less electronegative atom, what are they doing first off? What kind of bond do they share?
Covalent.
It's a covalent bond. And that means what exactly? Sharing valence electrons. So they're going to share valence electrons. Are they going to share them equally or unequally? Well, in the scenario I just gave you, it was with an electronegative atom with a less electronegative atom. So in that case, are they going to share them equally or unequally?
No.
Unequally, right? No. We don't have any of that going on. It is going to be shared unequally. So we've got unequal sharing then occurring with that nitrogen and the hydrogens that are on them. Because the nitrogen is electronegative. Not as electronegative as oxygen, but definitely more electronegative than hydrogen. Electronegative, what does that mean again?
The tendency to attract
more valence.
Yeah, so it's the tendency to attract those valence electrons. So if nitrogen is more electronegative than the hydrogen that is on it, or covalently bonded to it, then which of them is going to have a slight negative charge? The nitrogen or the hydrogen?
The nitrogen.
The nitrogen will end up with a slight negative charge, and that means that hydrogen has the ability to then hydrogen bond. It's a polar arrangement, and it can then hydrogen bond with a double bonded oxygen somewhere else. Now, in order to kind of illustrate this idea of what that can look like in a secondary structure, I want you to get a pencil. Some of you have already done this, and that's great. And I want you to take about half of your pipe cleaner, and I want you to wrap that half around a pencil or a pen. Okay, with your beads still evenly distributed around it. Okay, so just half of it, till it's curled around that pencil or pen, and then pull it out. and you'll be left with like a little curlicue, like a curlicue ribbon, okay? Now in a protein, we're going to call this curlicue ribbon an alpha helix. So it's a helix, like DNA, but it's not a double helix. So in a protein, we would just call this an alpha helix. So it's like a curlicue. Now this curlicue is the result then of hydrogen bonding between the nitrogen, so basically the amino group and the carboxylic acid group portion of the amino acids in different locations, not within the same amino acid. So, and you can kind of see how that can work. So your beads are distributed along your curlicue, and you can then imagine how they could be hydrogen bonding with adjacent amino acids, maybe two amino acids away, or even 3 amino acids away. You get that kind of curlicue. That's not the only kind of shape you can get for a secondary arrangement. So on the other half of your pipe cleaner, I want you to bend it like a series of V's. Like a Charlie Brown's, you know how Charlie Brown has that little squiggly sweater thing? And you can put your amino acids, you can distribute those like at the tops or of each of the little triangles that you would have. Now what you can then show, does anyone else have any triangles? Ooh, amino acids on the loose. Oh, you got it, good job. Does anyone else have this kind of squiggle yet? Because what you can do, you got a bunch of squiggles. Oh no, I see, that's a curly Q.
Yeah, this is.
Yeah, can I borrow your peanut bars? Okay, so what that can do then, remember, oh, how about this? How many amino acids do we have on these pipe cleaners? We have 15, right? Now in a protein, you could have 1,000 amino acids. So when we're looking at 15 at a time, realize that whatever we have here, I would have to divide 1,000 by 15. I can't do that in my head. You'd have a lot more than this, right? So what that means is that long chain of amino acids can wrap back around a bunch of different times. And so what can happen then with this kind of, this crunchy sort of fan type shape that you're making over here, if another strand comes along right by it, it can end up hydrogen bonding with the adjacent strand. And you can have a bunch of those together in what looks like ultimately like a fanned sheet of paper. And so we call these beta pleated sheets. Pleats, you know, like being in a fan. Makes sense. So this kind of alpha helix and this kind of beta sheet then that you could get, those are examples of secondary protein structure. And what I'd like you to do is I'd like you to draw then a picture of what your pipe cleaner looks like in the box marked, draw the secondary structure of your protein model. Now, once you've finished drawing that, yeah?
Do you have to have the exact same number and color and that kind of stuff written, or?
No, in the drawings, no, you don't. Yeah, it can just be pencil drawings. It doesn't have to be super advanced. Just enough for you to kind of remember what it is that we're talking about here, okay? Now, for the next level of protein arrangement, let's go ahead and,
So anything that I filled in,
could
you
you
can check that.
guys?
Oh.
Gavin, can you tell me about the next level of protein?
So it's ternary structure, and we're basically become like 3D.
Very good.
Complete.
So this is already kind of 3D, right? But it's not like super duper 3D yet.
It's not like
Yeah. So tertiary then, that tertiary level is when we're really kind of getting into that ultimate structure for that protein. And what I want you to do to model this then, we're going to have to get rid of our alpha helix and our beta sheets. Go ahead and straighten. Ooh, I just broke my protein. Straighten out your model. And what I want you to do now is you're going to need to follow the directions that are given in the procedure under number 4. So look at the procedure, and where it says number 4, the tertiary structure is based on side chain chemical interactions. Fold the pipe cleaner in the following order. So you already know that we've got 5 different colors, right? So let's go back to that pre-activity question where it said using the amino acid color chart below. This is number 3 in the pre-activity questions. What are the 5 different properties of amino acids? Nick, what do you have for the different colors?
Yellow is nonpolar, white would be polar, green would be sulfite bonds, blue would be basic, and red is acidic.
Very nice. So we've got those different, and where are those different chemical natures coming from?
The R group.
Yeah, this side chain, so that side chain interactions. So just like we were looking at the amino group and the carboxylic acid portion of the amino acids themselves, forming hydrogen bonds for the secondary structure. In the tertiary structure, it's all about what's happening with these side chains then. So in the side chains, that's what you're illustrating with the different colors with the beads that you have on your pipe cleaners. So what we're gonna do is we're gonna think about some of these different bonds. Now the strongest bond that we can have is gonna be a sulfide bond, which forms something that's called a disulfide bridge between 2 cysteines. Now, can anyone tell me what color cysteines are?
Green.
They're green. So what that means is with your protein right now, I want you to find your 2, you should have 2 green beads, right? And you're gonna take those 2 green beads and you're gonna take your pipe cleaner and you're gonna twist them together, right where those 2 green beads are, okay? Because that's what a covalent bond is gonna do. Can you just dissolve a covalent bond by heating something up usually? No. So if you boil water, it's not like all of the oxygen and hydrogen is just released into the atmosphere. It gets released as water vapor maybe, but it's not going to break those covalent bonds. What kind of bonds can you disrupt with heat like that? Hydrogen bonding. So a covalent bond is going to be way stronger than hydrogen bonding, right? So that sulfide bond is just a covalent bond that happens between cysteines. Cysteines contain sulfur. The 2 sulfurs then in the cysteines are able to form that covalent bond. Next, we're going to find any acidic and basic amino acids and we're going to pair them together in a 1-to-1 ratio to neutralize them. So when we're doing that, what colors are we looking for?
Red.
Red with blue. Now you've got 2 reds and 2 blues. That means you've got 2 different possible ways to pair them. So figure it out. I'm not telling you which way it needs to be. In this case, it's not like there's a right or a wrong. Is it going to be easier to pair the 2 that are super close together in your little loop now? Or is it going to be easier to kind of fold back over and pair them almost like pretzel style? But go ahead and twist those together. It's red with blue, not red with red. Red with blue, twist them together. Don't get rid of your green twist. You still need that. So, very good. So you've got your reds and blues twisted together, right? Now once you've got your acids and your bases paired together, you're going to need to fold the hydrophobic. Now the hydrophobic ones are yellow like butter, okay? So you're going to fold the hydrophobic amino acids to the inside in order to keep them away from the water-based cytoplasm. And then that means any polar ones. It also means that the acid and bases and the sulfide bond, so those can all be on the outside, but the hydrophobic ones have to get buried on the inside. So keep every twist that you already have and then get those yellow beads buried in the middle. And remember, you're not supposed to move them along the pipe cleaner, all right? They're staying all spread out like that. Uh-huh.
You brought pancakes.
I should have pretzels. I should have pretzels for protein folding. Yeah, you're not supposed to be getting all of them like that. They're supposed to stay distributed.
You bend it? Yeah, let's do it on the inside.
Now, hold up your tertiary structure then once you have it done, and realize it should not be flat. This isn't, we're not flat Stanley. We're not like all just two-dimensional in that regard. We're trying to get a three-dimensional globular kind of shape. So try to get it as much as you can. Who remembers flat Stanley? Right? Is that like, I was trying to think of that kid in her first grade. All right, so who has an example that I can potentially use?
Dude, ours looks so
Yours
good.
is pretty loose and open. Mine are kind of squished a little bit. I have it here. Oh, I see. These came apart. Okay, you gotta kind of twist your legs.
I got it.
All right, I'm gonna do a little tiny bit of this one if you don't mind. So the pipe cleaners, you can kind of twist them around. It looks like some of the chenille's coming off those pipe cleaners. All right, so chenille, that's the name of the fabric. Pipe cleaners. It's funny, like chenille sweaters. I love chenille sweaters. They're so soft. All right. So basically, okay, your pipe cleaner keeps breaking. So basically, you shouldn't be able to see those yellow ones very well. Okay, so the green ones should be twisted together, all the way together, and then the yellow ones should be on the inside, and then you should have all the other colors, so blue and red and green and white should be around them. And it's okay, guess what? It's okay if your protein ends up being kind of small. That's all right to have a small protein like this. It's not like it's filled. You know what I mean? It's not, I mean, if you think about it, we're just talking about what is going to fill the space, guys?
Water.
Water, right? Maybe some oxygen. Okay, you might have some gas. But we don't typically have voids. Why not?
Because you just don't.
Because you just don't, all right? All right. So this would be an example of tertiary structure then. Okay, that's what you're supposed to draw in the box marked, draw your tertiary structure of your protein model. So this is what you're going to have, is you're going to have that kind of crunched down version. I'm going to borrow this if you don't mind. Yeah, can I borrow yours too?
Look at ours.
A white bead.
Yeah, you're like right over there.
Yep, see?
I'm not sure who it is.
It's ours. It's ours.
That's protein.
So I need two more proteins if I'm gonna, I mean, 2 more polypeptides.
We got one. Here you go.
I love that. That's perfect. Your yellows are buried totally. That is beautiful. No. Thank you. Oh, that's okay. I've got 4 now. So here's the thing. So quiet down. Remember, it's hard to hear. So so this would be one example of a tertiary structure, right? So notice it is 3-dimensional. It's not flat. And it's kind of globular. Now, some proteins, that's all they have. They are now pretty much ready for any kind of final processing. And so they would be good to go at this point. Some proteins, though, however, do require additional polypeptides to actually make the final version of the protein. Hemoglobin is a good example of that. Hemoglobin has 4 different polypeptides that make up that protein, and they also are associated with a heme group, so they have an iron-containing group as well. So if we had 4 polypeptides ultimately making the quaternary structure, you would end up basically having a glob, so like so. Now in this case, I've got let's see, 15 amino acids times 4 polypeptides, so I've got 60 amino acids total. Again, keep in mind, a single polypeptide could have 1,000. So even if we were looking at one that had 600, it would be 10 times the size of this little pile right here, if we were trying to have like a real example. Oh, that would be fun. We should try that sometime. Like
We
just
got
get
time, we got time.
every pony bead that has ever come in a package. We're gonna get them all together on like the longest pipe cleaner ever.
There's a lot of amino acids.
That would be a lot of amino acids.
That's a
So-
lot of damage.
Anyway, so that would then be what you're supposed to draw for your quaternary structure. So in the box where it says draw the quaternary structure of your protein model, that's what it's looking for. Whose did I borrow? Borrow yours? You might not get the same
I
one
don't know what
back.
was so wrong with it.
Who else's did I take? Yours? Again, you might not get the same. Did I take yours? No. Whose else? Who else's? Did I take yours? Okay. It might not be yours. I'm sorry. Okay, so I want you to take a look now. Okay. So I want you to take a look at your polypeptide that you have in your hand right now. And now I want you to think about what a mutation is. What is a mutation? So I don't think we finished describing, by the way, quaternary structure. Nina, first of all, why don't you tell me what a quaternary structure is that we just modeled?
It's like a 4D structure and
Yeah, not totally thrilled with that definition. Let's go with another one.
When there's more than one polypeptide chain.
Yeah, it's when you have more than one polypeptide chain ultimately making up that final protein. Do all proteins have quaternary structure?
No.
No, they don't. But when they do, they have to have all of them there. And then what kind of protein helps with the folding of those kinds of polypeptides? So helping this arrangement to happen or helping that quaternary arrangement, what are they called? No. Chaperones.
Oh, yeah.
Chaperones. Yes, you do have hydrogen bonds, but no, the proteins that are actually going to be helping with this, you know, so like our hands essentially, right? That would be, our hands would be acting as chaperones or chaperonins. So this is what I want us to think about now then. What is a mutation? So Niharika, what is a mutation? It's not. Okay. Okay. All right, we're getting close. Let's say it does have to do with nucleotides, but on what kind of molecule? Because this is really important.
Is it just an error in the genetic code?
Not the genetic code, no.
When the RNA, like.
Not RNA.
DNA?
Yep. Exclusively. Mutations take place in DNA. And it's not that there's something wrong with the nucleotide, it's that the wrong nucleotide is inserted. So let's say that you're supposed to have an adenine. And instead of adenine, a thymine is put in its place, or a guanine or a cytosine. Then in that case, we would call that a mutation. And the mutation then exists exclusively in the DNA, but it does mean that a messenger RNA, an mRNA that is transcribed from that DNA may end up containing a codon that codes for a different amino acid, but will it always be a different amino acid?
No.
No. Take a look at your genetic code for a moment. So what pages are those on again? I think it's like 248.
238. 238.
238 and 242. So take a look at your genetic code. Now, and that's why I was saying no to the genetic code, Gabe, because the genetic code, that's what we use with mRNA. And so it's not considered to be a mutation. The mRNA is just going to be a copy of the DNA. So the mutation would be in the DNA itself. Yep.
So I'm confused on what the names, like, what do the names mean?
For what, amino
For
acids?
the genetic code, like, so those are the amino acids.
Those are the amino acids, yeah. So like alanine, all of that, guys. So alanine, all of those kinds of names you're talking about, those are the names of the amino acids. And so each amino acid that has a different R group is going to have a different name, okay? So I want you to look at your genetic code for a minute, okay? when you're looking at, let's say, find the codon UCU. Okay? UCU. What amino acid does UCU?
Serine.
Right, serine. So in the DNA, UCU would be what series of nucleotides would UCU be in the DNA? It would be AGA. It would be AGA. So UCU, if that's your codon, then the DNA that it was transcribed from would have been AGA. All right, and so we could look at the DNA. Now, what happens to UCU if the DNA, that's where mutations take place. If the DNA were to be so instead of AGA, it was AGT. What would that do to the codon? So yeah, if we had AGA, which makes the codon UCU, which codes for serine, right? If we had a mutation and it was no longer AGA, but it was now AGT, what does that do to the codon?
UCA.
All right, so we went from AGA in our DNA, which makes our mRNA UCU, which makes our amino acid serine. What happens then if we have a mutation right here? And so instead, we had AGT. What's our mRNA going to be now? It's going to be UCA, right? And so what codon is that, what amino acid is that codon going to code for now? What? Okay, so that's going to be serine. So do we see any change? Is the protein going to be affected at all?
No.
No. Did we have a mutation?
Yes.
Yes, we still had a mutation. We went from A down here to T. We definitely had a mutation. In those cases, we call those silent mutations, when it doesn't end up changing anything at all in the actual amino acid. Yep.
Is that because like they both they go with each other, right? Like A and T go with each other. So like if it was like
They
C or G,
complementary base pair. Yeah.
But if it was like C or G, would it change?
What do you? Oh no. You mean going from A to T?
Yeah, like if I had
No.
a
Okay.
different
How about we
mutation.
change it? All right. Here, we'll change this to G. How about that?
Okay.
We change that to G. What is it going to? Then the mRNA is going to be what?
Oh, so
UCC. What does UCC code for? Still coding for serine. All right. And this has to do with this kind of redundancy. If you notice on the genetic code, when you look at it, there are a lot of amino acids that are coded for by more than just one codon, right? So serine has 4 different codons. Oh, 6 different codons. Look over there. It's also AGU and AGC. Serine has 6 codons, all of which code for serine. So you can change certain aspects in the DNA. You can have mutations, and it doesn't even necessarily mean that you're going to change the amino acid. But let's think about what happens if it does.
Well, going back to the previous where it's a silent mutation, is that sort of like a fail-safe that the body developed, or is it just luck?
Everything in biology is luck. Yeah, it's all luck. Yeah, that would be cool if we were like, hey, we need to design a better body, and then we like designed a better body.
Can we do that?
Oh no, no.
Could we train
Yeah,
our, like,
evolution.
ourselves?
Yeah, evolution through natural selection ensures that no, it's basically you luck out and you get something really cool and you survive to reproduce better. But no, it doesn't what kind of cool bodies would we have if we could do that? I would have wings for sure. I would totally be flying around places. I would also be green because I'd want to photosynthesize. Who wants to bother eating stuff if you can just like photosynthesize? So I'd be green and flying. Oh well. Genetic engineering at its finest. Let's go for it. All right. So let's go ahead then. So this would be an example of a mutation that doesn't end up affecting the protein at all. Still a mutation because mutations happen where?
In the DNA.
In the DNA. In the DNA. So what if, let's take a look at our little list here, our genetic code. What if instead of having a mutation here, what if we had a mutation, so we're still talking about our serine, right? What if we had a mutation in the DNA to look like, how about if this A were turned into a G? So what if we went from AGA to GGA?
Different.
What is our mRNA going to look like?
CCU.
It's going to look like CCU, so it's still dealing with just one single point mutation, one spot, but instead of that last spot now, we're looking at mutating that first position. So now we have CCU. What is CCU code for?
Different amino acid.
Really?
What's the amino acid now?
Proline.
Proline.
Proline, yeah.
Okay, I want you to look at your color chart and tell me what color is serine and what color is proline. Serine is white.
Yellow.
And proline is yellow. So what does it mean if you're a white bead?
Polar.
Right, so polar. What does it mean if you're yellow?
Non-polar.
Uh-huh. What did you do with your polypeptide? So hold up your polypeptide. I already straightened mine out. So hold up your polypeptide. What did you do? What did you have to do to your yellow beads?
Tuck them in the middle.
Your
Shove
hydrophobic
them in the
beads.
middle.
Why did you tuck them in the middle?
Because they don't like the polar outside water.
Because they're trying to stay away from the very polar cytoplasm. Remember, we're mostly water. So that water-based cytoplasm then, if you took one of your white beads and you turned it into a yellow bead, or you took one of your yellows and turned it into a white, whatever, would you fold it the same way?
No.
There is no way that you could have the same exact folding that you have right now if one of your white beads were now a yellow bead. It could not fold the same way. This is important because one single point mutation that ends up changing an amino acid with one chemical characteristic into an amino acid with a different chemical characteristic can change the structure of the protein. Structure determines function. Is that protein still going to function normally?
No.
No, it can't. And if this happens in an active site of an enzyme, is it still going to even be able to catalyze a chemical reaction?
Maybe the wrong one.
Yeah, maybe the wrong one. Sure. But it's not going to catalyze the one that you want. Or it's not going to do it very well. This is an example, or this is seen as an example with sickle cell anemia. In sickle cell anemia, you've got one single point mutation that ends up having a change like this, and it causes hemoglobin to fold abnormally. What does hemoglobin do?
Transfers oxygen throughout the
It's carrying oxygen around to your cells. Again, final exam in 2 weeks. Why do your cells want oxygen?
For cellular.
Because it's the final electron acceptor of the electron transport chain of cellular respiration, which is going to produce what?
ATP.
ATP, which is what?
Energy.
Energy. So guess what? People that have sickle cell anemia, guess what? They're lethargic. They don't have a lot of energy.
That is so
They
sad.
get tired very easily. They can also faint because they don't get enough oxygen to their brain cells, and it can cause them to have fainting episodes. There's a lot of other issues that happen. The hemoglobin causes the red blood cells to have like this really weird kind of spiky shape, and that means that when they're trying to go through capillaries, they sometimes get clogged. So that causes a whole other set of issues. All of it the entire disease of sickle cell anemia caused because of one single mutation in one single nucleotide in the hemoglobin. And that causes everything to happen that we see. It also explains genetics, because when you have two bad copies of that particular allele, then that means you can only make that malformed kind of protein. But if you have one good copy, you're still going to be making some good hemoglobin. We'll talk about it again in genetics, yeah.
So are like the silent ones, the silent mutations, are those like kind of common?
Yeah,
But then are these ones less common?
yeah. Because going back to that whole idea of natural selection,
Yeah.
if you end up destroying the functioning of a protein that's really important, then you generally aren't going to be as successful in surviving to reproduce. And so really bad mutations that really mutate proteins tend to cause less fitness of an individual because they don't survive long enough to reproduce.
And could like one mutation like that like damage the whole body?
Sure, yeah, because that one mutation is going to show up in every single one of your cells. Because that's you've got, if it's in the master copy of your DNA, that means the child would be born with it, then that means every single cell is going to have that mutation in place. And so, yeah, that means it's system-wide that the effects are going to be felt. And of course, it depends on what the protein does, right? So what effect do you have depends on the protein. If you look where it says that Human Genome Landmarks poster that's over there, most of what you see listed are genetic disorders. Those genetic disorders oftentimes are going to involve things like single point mutations like this that cause a malfunctioning protein. And it ends up causing a certain genetic disorder or genetic disease that, that we can list out, map on the chromosome and say this is why you have that, not that it fixes it. But at least we know why. Yep.
So if the mutation first happens in only one cell, would it take a while for it to happen throughout the whole body?
It might not happen throughout the whole body. So what happens, it's not like cancer. It's not like a cell that has one mutation in it is going to be able to spread throughout the whole body. So could you have, one single cell that ends up with a mutation that makes one malformed version of a protein? Sure, you could. Is it going to affect a whole lot? No, it's not. Because how many times does that single cell reproduce? How much protein does a single, think about it, they're invisible. That's so weird. But how much can a single invisible cell actually affect in its one little area? Not much. So the big problem is when you have these mutations from birth. So that means in the original egg or the original sperm, if there's a mutation there, that's when we have to worry about it being a systematic or system-wide genetic disorder, not just if it's one cell or two even. I mean, we're made of trillions of cells, so it's like a drop in the bucket. Yes?
Okay, I was wondering if we have like an actual cure, permanent cure for all these things, like a lot of disorders like this,
No.
or yeah, we have suppressive medicine, right?
You're going to suppress the production of
Well,
hemoglobin.
no, I mean like we have
I don't
ways
think that's a good
to
idea.
stop the issues, but like you don't have an actual cure. Like temporary.
Again, how do you stop it? I mean, what can you do? Yes, we have ways of treating certain genetic disorders, but no, we don't have cures for most genetic disorders. Gene therapy is one avenue that they're attempting, and they're trying to use things like some CRISPR technology, which is kind of scary really because we don't really know what the side effects are going to be. If you go in and start trying to change people's genes, how good is that going to be? I don't know. But I mean, for people, if you've got a deadly disorder, it might be worth trying. Just dealing with whatever the side effects are. But yeah, that's not generally. You do things to try to make the person more comfortable whenever possible. But you can't go in and fix all of the trillions of cells that have a particular point mutation in it. Again, I think they're trying to do some things with CRISPR, which is pretty interesting, but I don't know how successful it's going to be. It's not super successful yet. All right, so that whole discussion was to help you with the post-activity questions, in particular numbers 4 and 5. So I'd like you to go ahead and I'm going to give you just a couple minutes. I'm going to pull up a Bozeman video for us to watch, and I'll give you a couple of minutes to try and finish up, at least look at those questions and determine what you think the answers would be. Yes, please. If you could also please disassemble your pipe cleaner and beads and put them back into the baggie, that would be great. I'll pull up your Bozeman video.
You put up, I know, every other.
I lost my Bozeman video. Okay. Guys, remember I passed out that Bozeman video to you earlier for you to work on the back side. Please get out your Bozeman video worksheet so that you can answer those questions now.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about transcription and translation. In biology, that's how we go from DNA to RNA, and then from RNA to proteins. And so we are made up of proteins. In other words, we eat food, and that food is going to be reassembled to make us. In other words, you are made up of proteins, and those were made up of amino acids. And so just like when you start to cook pizza, and if you want to cook pizza, you're basically going to have to get all the ingredients together and then put those together in the right order to make a pizza. Same thing works inside you. The DNA is going to tell your body how to organize the amino acids and into those proteins, and those are eventually going to make you. And so if we talk about that cooking analogy, basically the cookbook, if we're cooking in the kitchen, is going to contain a bunch of recipes to make a number of different foods. The chef is going to pull the right recipe out, or you could copy it down on a little index card, and then you bring it to the kitchen where you take all these ingredients and then you put them together correctly to make pizza. And so if we switch to the next slide, what's been replaced in our kitchen? Well, the cookbook is going to be like the DNA. It sits in the nucleus, it's protected. The chef has been replaced by a number of different ribosomes. The recipe is now messenger RNA. And if I were to do this accurately, I should have hundreds and hundreds of different index cards. One copy of the DNA in a cell, but hundreds of copies of the messenger RNA. What are the ingredients then? Those are going to be the amino acids, and we're going to put those together in the correct order, and then we're going to make a protein, and that protein makes you. This process that I'm describing is called the central dogma. And the central dogma was developed by this man, Francis Crick. So after Watson and Crick came up with the structure of DNA, he spent years working out how this actually works. And so the process he called central dogma. Basically what we do is we take the DNA. DNA is going to have sections of it, which are called a gene. And the gene is going to code for a specific protein. And so basically we'll copy down that gene into messenger RNA. And that process is called transcription. Transcription is going to take place in the nucleus. And so all of this up here is going to take place in the nucleus. And this down here is going to take out in the cytoplasm. And I'm talking about eukaryotic cells like you. So basically we'll copy down that gene into messenger RNA. We'll manipulate it a little bit. We're going to modify it a little bit. But basically, once we've done that, then that messenger RNA is going to move out through one of these nuclear pores. It'll grab onto a ribosome, and then it's going to make these things, proteins. Proteins, that process is called translation. And so basically, the way I remember it is script in transcription stands for to write. And so we're writing down the message of the gene into messenger RNA. So it's a message. And then that messenger RNA is going to go out here, and we're going to have a number of amino acids that weave together, and then we're eventually going to create something like this, a protein. Now what does that protein make? That protein makes you. In other words, you're made up of a bunch of different proteins. Where's the recipe book for the proteins inside you? That's going to be in the DNA in the nucleus of every one of your cells. Okay, so let's get to more specifically how that works. If we're talking about eukaryotic cells, remember all of our DNA is going to be contained within a chromosome. And so that chromosome is a bunch of DNA that's wadded up, or it's wrapped around histone protein. So if we unwind it, you can see that double helix. But if we really unwind it, what you'll find is there's going to be a message on either side. This enzyme is called RNA polymerase, is going to move down that DNA, and it's going to copy the information in the DNA. And so if we've got a C here, that's going to be a G here. So we're going to have complementary bonding, and we're going to create the transcript. It has the same information as the information that's found in the DNA. And then we're eventually going to lose that RNA. And now the nice thing about DNA is it can zip together because there will be hydrogen bonds between the two sides. And now that gene is protected within the DNA. So if we go through that in the steps of transcription, this would be RNA polymerase. RNA polymerase is going to grab onto the DNA. There's a number of transcription factors that are going to allow that to occur. Here's our gene. Then as the RNA polymerase is going to move down that DNA, you can see that it's creating a copy behind it. And that copy is called messenger RNA. So this is our messenger RNA. And if you look, as RNA polymerase takes off, we've left the message behind, and the DNA zips back together again. And so we have this initiation, elongation, and then finally termination and the creation of that messenger RNA. Now in us, we're going to do some things to it. We're actually going to put a 5' cap on one side. We're going to put a poly A tail, which is just a bunch of adenines in a row. Another thing that we'll do inside us is that we're going to get rid of introns. Introns are sections of that don't actually code for a gene, so we'll get rid of those and then splice together that messenger RNA. But basically what we have when we're done with transcription is some messenger RNA. Where does that messenger RNA go? It's going to go outside of the nucleus and it's going to go into the cytoplasm. What's going to occur there, that's where we're actually going to take that recipe, or we're going to take that message, and we're going to make a protein out of it. We're going to build that out of amino acids. Okay, so how does translation work? Let's kind of orient ourselves, because this diagram seems crazy. Here's our messenger RNA. So this is going to be our messenger RNA right here. And so it's moving through a ribosome. So a ribosome is where we make proteins. It's going to have two parts to it. It's going to have a small subunit, that's this bottom part. It's going to have a large subunit on the top, and then the messenger RNA is going to flow right through it. Every 3 letters in the messenger RNA is going to code for 1 amino acid. And the reason why is since we have 4 letters in DNA, 4 letters in RNA, A, T, C, and G, or A, U, C, and G, if we're talking about messenger RNA. Every 3 letters are going to code for 1 specific amino acid. And the reason it's every 3 letters is it gives us enough variety that we can cover all the amino acids. So speaking of amino acids, where are they? The amino acids are going to be these little balls, and they're on top of another type of RNA that's called transfer RNA. Transfer RNA's job is to transfer amino acids from the cytoplasm to the ribosome, so we can attach them on to this growing polypeptide chain, which is going to become this protein. And so basically, what does a transfer RNA do? Let's just kind of look at one. It's going to grab its amino acid right here, and then when it's its turn to go to the ribosome, it's going to bond to the ribosome like this, and then it's going to drop off its amino acid. And so it's basically dropping off this amino acid. It's then going to flow out into the cytoplasm to grab the next amino acid. Now each tRNA is going to grab a specific amino acid, and it's going to have 3 letters on the bottom of it. We call.
Okay, I'm going to pause him for just a second because he's talking about dropping off amino acids. But I want you to think about it in terms of chemical reactions. So what chemical reaction is taking place between the amino acids that are being dropped off by the tRNA? Dehydration synthesis. So we're having dehydration synthesis taking place. So that is what is actually connecting each of those amino acids. So yeah, the tRNA comes in, it drops off the amino acid, which is going to, through the process of dehydration synthesis, covalent bond with its neighboring amino acid.
Call that the anticodon that's going to bond to the codon that's found in the messenger RNA. So basically this occurs over and over and over and over again until it gets to a stop sequence on the end, and then we're done with that protein. And so I'm going to start this animation over here, and so basically let me kind of talk you through it. This down here is going to be the small subunit. This on top is going to be the large subunit. And then we can see that we have all of these, they're blue, these dark blue, are coming in and they're actually bringing their amino acids with them. Now there's an enzyme that attaches that on. But basically these dark blue tRNAs are each dropping off an amino acid, and then they're taking off into the cytoplasm to pick up another amino acid. You can see right here that the messenger RNA that came from the nucleus is just feeding through the middle of that ribosome. And it's a site where we can actually have the tRNA come in. Now, if we watch this animation here, this is a ribosome that's bound to the endoplasmic reticulum. And so it's going to attach to the endoplasmic reticulum, and that growing protein that was growing inside that large subunit is actually going to grow into the middle of this endoplasmic reticulum. And so we can modify it or do things with it. And so basically this is translation. So translation, if we go back to our analogy again of the cooking analogy, basically the ribosome serves the purpose of the chef. And so it's taking together all these different ingredients, all these different amino acids, and putting it together to make a protein. So we're translating that message into a functional protein. You can see the protein coming out on this side. Okay, what I'm going to try to do here is actually decode a gene. So we've got gene found within this stretch of DNA. So this is a double helix, so we have complementary strands. And so basically what I'm going to do is the process of transcription. So I'm going to go from DNA.
He's usually really good about that kind of stuff, but in this case, that's just wrong.
DNA to messenger
Do you ever
RNA.
have you in a
And then I'm going to
DNA?
try to translate that
Never.
message, and I'm going to go from messenger RNA, or through the process of translation, I'm going to go to the amino acids found in the protein. Okay, so basically, I'm going to play the role in this first line of RNA polymerase. So in the cytoplasm, RNA polymerase is going to look what's at the DNA, and it's going to put a complementary RNA. So in the DNA, A goes to T, but when that unwinds, the T will actually go to A in the messenger RNA. T goes to A goes to U. Remember, we only have uracil in messenger RNA. A to C goes to G. So if I just keep going, this right here would be an A and a C.
He doesn't figure it out,
This is
ever,
our
even though he just said that.
Yeah,
G.
He looks very tired.
This is C.
I think he's pretty tired. He
always
also,
looks tired.
And then this is
Well,
going
that's
to be
just
a U and an A and an A. And so every messenger RNA
right
is going to
there.
be what we call a codon, because it codes for a specific amino acid. Now if we want to do the actual translation and figure out what amino acids are created, well, I can't do this from memory. I have to use one of these standard genetic code decoders. Some of them be circular, some of them be like this. But basically what you do is you look at the codon in the messenger RNA, so AUG, and then I just find that down here. So here's AUG right here, so that's going to be methionine. So methionine is going to be the first amino acid. Now, methionine is special because it also starts all genes. All genes are going to start with methionine. Sometimes we trim that off later, but basically that's going to be our start sequence. Let's go to the next one, ACG. So I find ACG. This is threonine, so that's going to be like this. If we go to the next one, it's GAG. So GAG, that's glutamic acid, so that's going to be GLU. If we go to the next one, it's CAA. So let's find that CAA, that's glutamine, so that's going to be GLN. And then our last one is going to be UAA, so when I find that, you can see it's right here, UAA, that's going to be a stop sequence. And what does that mean? Well, that's the end of the gene, so that's not going to actually code for an amino acid. It'll actually put in a release factor so the whole thing can break out. And so this is going to be our first amino acid, our second amino acid, our third amino acid, our fourth amino acid. This whole thing then is called a polypeptide. And again, it would fold into a specific protein to do a specific job. Now, most of them are going to be much longer than this, but what I've shown you is how DNA becomes messenger RNA, amino acids will eventually form proteins, and now that whole thing is eventually going to make you, because you're made up of proteins. And so that's transcription, that's translation, and I hope that's helpful.
All right, and I hope it's helpful too. Okay, guys, the bell is gonna ring in a moment.